Since this is the first video podcast that we've really done for this class, I want to remind you that the better way to actually look at these podcasts is by doing the projection mode that you can do at the bottom left-hand side of the screen. We tell you when you need to animate the slides, so if you click on them when we tell you, it will actually pop up, and that will help you work through a little bit more of the complicated slides.
In this lecture, I'm going to introduce you to some new concepts related to Mendelian inheritance and teach you how to work through problems that allow you to use your understanding of Mendelian inheritance and meiosis to calculate the probabilities of certain offspring occurring, the frequency of expected genotypes and phenotypes. With this, you'll be able to tackle all of the problems in the spreadsheet or the worksheet that we've prepared for you to go with this lecture, and you'll be able to handle all of the questions on your final exam. We're going to start out by working through a very simple problem. This is one that's probably something similar to what you've done in the past and what you've done in the lab. So here you can see Vinny. This is Dr. Simula's cat. He's all dressed and ready to go to the zoo to see all the other cats. And you'll notice that Vinny is a bit unusual. In this photograph, you can see that Vinny actually has 6 toes instead of 5. Where the dew claw is, higher up on the paw, he has 2 claws. And this clearly indicates that he has a condition known as polydactyly. Okay? Polydactyly is a trait that is exhibited in individuals carrying at least one copy of a dominant allele. Okay? So this mutation of this gene that confers 6 fingers and toes is dominant to the version of the gene, the allele, that codes for the normal 5 fingers and toes. Okay? Now that you know Vinny has 6 toes, what do we know about him? Do you know his genotype? The answer to this is no. You do not know Vinny's genotype, and that's because polydactyly, being a dominant trait, Vinny could be heterozygous. He could have one dominant 6-fingered allele and one recessive 5-fingered allele, but because polydactyly is a dominant allele, he has 6 toes. He could also be homozygous for polydactyly. Okay? If he were homozygous for polydactyly, had 2 copies of that 6-fingered and toed allele, that would also tell you something about Vinny's parents. I invite you to think about that on your own, and we'll talk to you about that in lecture next week. See whether you come to the proper conclusion regarding Vinny's parents. Now I'm going to introduce you shortly to Matilda, Dr. Simula's other cat, a female. If Matilda and Vinny have babies, could that help us determine whether Vinny is homozygous or heterozygous for the gene responsible for polydactyly? If they had enough offspring, it certainly would. If Vinny were homozygous for the gene that confers polydactyly or determines the number of toes, if he's homozygous for that, and Matilda, who has 5 toes, is obviously homozygous recessive, I encourage you to draw out the possible Punnett square, and you would conclude that all of Vinny's offspring would have polydactyly if he were indeed homozygous for this dominant allele. If, on the other hand, Vinny is heterozygous and Matilda, we know her still to be homozygous recessive, in that situation, half of Vinny's offspring would have 6 fingers and toes, half would have the normal complement of 5.
Before we move on to doing a cross between Vinny and Matilda using a Punnett square, let's review what Dr. Noonan has already covered. First, Matilda has 5 toes, like you can see in the picture. And from that information, do we actually know her genotype? The answer is yes. we know that she is homozygous recessive for the polydactyly trait. Now, before we move on to make a Punnett square, we should always think about the types of gametes that each of the members of the cross can make. So, when we think about Matilda, what are the gametes that she can make? If we use the letter P to describe our trait, a capital P would be for polydactyly and a lowercase p would be for the recessive trait of normal 5 digits. Therefore, Matilda's genotype should be little p, and the gametes that she can produce are little p. For Vinny, things are a little different. Do we actually know any of the gametes that Vinny could make? Well, since he has 6 toes, we also know then that he has to at least have one big P. But we don't actually know what his other allele is. So if you remember, when we talk about trying to figure out what the genotype of an individual who shows the dominant phenotype is, we use a test cross. And a test cross is when we actually take a homozygous recessive individual and mate them to the unknown. So in this case, Matilda would be the example of the homozygous recessive individual. So now let's move on and talk about building that Punnett square.
Here I've illustrated for you the 2 Punnett squares in the scenarios I just described for you. The rows represent the possible gametes that Matilda can produce. She can produce eggs that possess the small recessive P that her mother gave her and the small recessive P that her father gave her. Either way, she can only give to her offspring the recessive allele for this gene. Vinny, on the other hand, has 6 toes and 6 fingers. So if you look at the top of the Punnett squares, at the head of the columns, on the left, I've illustrated how this Punnett square would look if Vinny were indeed homozygous dominant. In that case, he would give all of his offspring either the dominant P his mother gave him or the dominant P that his father gave him. His genotype being homozygous, that's all he can give. On the right, you can see if Vinny is indeed heterozygous, he has one dominant copy of the gene and one recessive copy of the gene. In that case, half of his offspring will receive the dominant P, the 6-toed P, and half of his offspring will receive the recessive P, conferring 6 toes. Okay? So on the left, you can visualize what proportion of the offspring will have 6 toes, and contrast that with the scenario on the right. In these types of scenarios, you can be given questions that ask you about if we see half the offspring have 6 toes, or if all of the offspring have toes, what does that tell you about Vinny? I want you to be thinking about these questions in the context of a Punnett square that helps you infer what the genotype of the parent in question is.
In the last example, we spent time talking about the idea of simple dominance, where the presence of one allele, the dominant allele, hides the phenotype of the other allele, the recessive allele. Not all alleles work this way. At some gene loci, you have 2 alleles, but you don't end up with complete hiding of the recessive allele. Instead, what you get is a combination of the 2 alleles together. When this happens, we can call this incomplete dominance. The key feature of incomplete dominance is that when you put the 2 alleles into one individual, so if you put a dominant allele, so in this case a big P, and a little p together to make a heterozygote, that heterozygote has a phenotype that's unlike either of its parents. In this example we have from with eggplants, what you see is a parental generation where you have a purple eggplant, and that genotype of the purple eggplant is big P, when the white eggplant is little p. When they make gametes, the purple fruit would produce only big P gametes, and the white fruit would only produce little p gametes. That's the same as you would expect to see with breeding individuals in a simple dominance example. The difference is when these two individuals mate and produce offspring, all of their offspring end up heterozygous. But these heterozygotes look nothing like the parent. Instead, there's some intermediate, or they have a completely different phenotype from the parents. In this case, they use the term violet fruit, but it's really just a lighter purple color. Those individuals then are big P, little p. If we then cross two individuals with the violet coloration and perform a Punnett square, what we see is that you will still see the same genotypic ratio that you would in a simple dominance example. You have one big P, two heterozygotes of big P, little p, and then one homozygous recessive of little p's. However, the phenotype ratio now matches the genotype ratio. You have one purple to two violet to one white. So the key thing here, when you see an example of incomplete dominance, you should expect to see the genotype ratios matching the phenotype ratios because each genotype has a different phenotype. You might be wondering how incomplete dominance might work. Well, one of the good examples that we have is in these flowers called snapdragons. Snapdragons come in a lot of different colors, but in the red, white, and pink individuals, we actually understand the biochemical mechanism that makes this work. To make things simple, we're going to talk about the genotypes of the plants first, and then I'll explain how we actually get red, pink, and white flowers inside the cell, from inside the cell. So when you have a red flower, you have the genotype big R. And when you have the white flower, you have the genotype r. So the red and the white flowers represent the homozygotes. The heterozygote, on the other hand, is pink. So it's sort of an intermediate phenotype, but it's a different phenotype between the two parents. Okay? This is not blending like we talked about with paint, but instead it's actually a combination still of the big R allele and the little r allele. If you do a test cross or cross a heterozygote with a white individual, you see that you still get some white offspring. So this isn't blending, this is still the same idea where we're taking two alleles, making an individual. Now, to make this work, we have to understand a little bit about how we make a pigment. So pigment molecules are the ones that give something color. So to make a pigment inside of a cell, you need a molecule to start with, and then you need an enzyme to modify that molecule to make the pigment. We call the molecule we modify the precursor. So in order to make the red pigment, you need the precursor molecule and you need the enzyme that modifies the precursor. In snapdragons, what's happening is not about the pigment itself. It's about the enzyme that makes the pigment. When you have two copies of the functioning enzyme allele, then you can make more red pigment. So red individuals that have the big R genotype also have two copies of the functioning enzyme. So they can take lots and lots of the precursor molecule and make it into red pigment. So the flowers appear really red because you can make a lot of the red pigment. If you have two copies of the little a allele, you cannot make a functional enzyme. It doesn't actually work to convert the precursor into the pigment. So if you have one good copy and one terrible copy like you do in a heterozygote, then you actually only make a little bit of the red pigment because you don't have enough enzymes to modify that precursor. What you can see in the table that I've shown you here is the phenotype of the flower, the genotype of the flower, and then the amount of protein or the amount of enzyme that you make. So an individual who's big R, makes 2 times as much enzyme as the one that's big R, little r. Now, this is only one way that this works, and it happens in a lot of the incomplete dominance examples. However, there are other mechanisms we won't talk about, but because you guys understand enzymes, this is one that you can actually understand how the mechanism works.
Okay, now we're going to move on to an example of a trait that does not obey the patterns of simple dominance that were observed in the pea plants that Mendel worked with originally. This is talking about the different human blood groups and blood types. You can see here we've illustrated all 4 types of blood cells: type A blood cells, type B, type AB, and type O. Now the type A and type B blood cells, those are determined by the antigens on the surface of the blood cell. So type A blood cells have an A antigen on the surface, indicated by these red lollipop looking structures. Type B blood cells, that their type is determined by the B antigen on the surface of the blood cell. But what you'll notice here is that individuals with blood type AB have both antigens on the surface of their blood cell. This, unlike incomplete dominance, is a case in which both alleles are expressed completely. Their individuality is not compromised by the presence of the other allele. They don't become somewhat in between the antigen of an A and a B. There are discrete and specific A and B antigens on the surface of those AB blood cells. Notably, you are probably aware that there are people with blood type O. Interestingly, this is a nonsense mutation that has led to the disruption of the production of the antigen, the protein that serves as the antigen on the blood surface. So individuals with blood type O have no antigens on the surface of their blood cells. Okay. So below, I've indicated the blood types and what genotypes can cause those blood types. And you'll notice that for type O, there's only one way you can be type O. If both of your versions of the antigen coding gene produce non-functional antigens, if they both have nonsense mutations, you will produce no antigens. So if you're homozygous for O, you'll have no antigens on the blood surface. The only way to have blood type AB is for one allele, one copy of your antigen gene to code for the antigen A, and the other copy of that gene to code for the B version of the antigen. Okay, so you must be heterozygous, possessing both an A and a B allele. Interestingly, individuals with blood type A or B could be homozygous for the A or the B alleles, but they could also be heterozygous, possessing one copy of, for example, the A allele, and also a single copy of the O allele. So here I've given you a scenario much like one you will see on the worksheet that you'll be doing later, or that you might encounter on an exam. In this, I've told you that a man of blood type A is married to a woman with blood type AB. When given scenarios like this, whether it's on a worksheet or a test, I want you to first determine what it is you do not know. Is there anything you don't know about the woman's genotype? No, you know everything about the woman's genotype because there is only one way to become AB. For the man, it's not yet clear. There are 2 ways to have blood type A. He could be homozygous for A, or he could be heterozygous, having allele A and allele O. So let's look at the questions I've posed to you. what phenotypes might their children have? If they pair we know that the man has an allele A that could pair with the woman's allele A, so they could be blood type A. We know that the woman has allele B. It's possible that the man has allele O. So if those two paired up, B and O, their phenotype might be type B. And if allele A from the man and allele B from the woman pair up, the offspring could also be AB. Now, an interesting permutation of this question would be for me to ask you, if they produce offspring, their first child, let's say, and that child's blood type is B, can you tell me the genotype of the man? Notably, of all three of these phenotypes, it's only phenotype B that allows you to tell me that that man's genotype is for certain AO. Because we know the only B could come from mom, the only way the offspring could be B is if they get an O from dad. Okay? Now, let's think about what genotypes their children might have. Okay? If dad is AO, then the possible genotypes are the three listed here, basically the ones that we touched on above. Okay, but these are the genotypes, and that is how you would get the B genotype, having the B and the O alleles. If dad is homozygous for A, then there are only two genotypes possible, the A homozygote and the AB heterozygote. Okay. Now, let's think about if dad is indeed AO. I could ask you, what proportion of his children do you expect to be type B? Now, to do this, it might be useful for you to draw a Punnett square, and then on the top, you can put the mother's possible alleles that she can give to her offspring. She can give her offspring A or B. You expect her to give half of her offspring A, half of her offspring B. The man, if he's AO, could give half of his offspring A, half of them O. Okay? In this case, how many possible combinations are there that produce blood type B? There's only one combination, the B and the O, that can produce a type B offspring. So for these two parents, if the dad is AO and the mother is AB, we expect there to be 1 out of every 4 offspring, or 25% of the offspring, with type B blood. This is the same as saying, or you can say, based on the same reasoning, that there are 4 possible genotypes for the offspring, and 1 out of those 4 possibilities is type B. All 4 possibilities are equally probable. Okay? And so a 25% chance for each child of being type B, and 25% of the children are expected to be type B. Here we're going to begin discussion of sex-linked traits, and these are traits that reside not on the autosomes, but on the chromosomes that contribute to determining the biological sex of an individual. For humans, we have 22 pairs of autosomes, chromosomes that are not involved in determining our sex, our biological sex, and 1 pair, the 23rd pair of chromosomes, that determine the biological sex. That 23rd pair for biologically female individuals consists of 2 copies of the X chromosome. For biologically male individuals, they possess a single X and a single Y chromosome. This is probably not anything new for you, but this is going to get a bit more complicated as we discuss the inheritance of traits that are the product of genes on either the X or the Y chromosome. So the humans that are male, biologically male, possess one X chromosome, one Y chromosome. Biologically female individuals possess two X chromosomes. Now, it is the Y chromosome that makes individuals male, not the absence of a second X chromosome. And as you look at how these are denoted on the screen, you'll note that you see XY next to male and XX next to female. Please don't confuse this with a replicated chromosome. This is more of a name than a depiction of the physical structure of the chromosome. Okay? So, in humans, genes that reside on the X or the Y chromosome, which of those could a biologically female individual be homozygous or heterozygous for? Okay. They could be homozygous or heterozygous only for genes that reside on the X chromosome. Since they have no Y chromosome, they can't even possess any genes that reside on those chromosomes. Now males, think about them and whether they are homozygous or heterozygous for genes on either the X or the Y. And you'll quickly come to the conclusion that they cannot be homozygous or heterozygous for any genes on either of the sex-determining chromosomes because they have only one of each chromosome. The term we use for males, in this case humans, males that are the heterogametic sex, they have two different chromosomes. The term we use to describe their state is hemizygous, H-E-M-I. Okay? So males in humans are hemizygous for traits on the X chromosome because they have but one copy of that gene. They're also hemizygous for traits due to or coded for by genes on the Y chromosome. They have but one copy. Now we're going to consider what happens when these two individuals that the male is hemizygous for a gene on the X chromosome that confers normal vision, can see all colors. The female in this scenario, she possesses one normal copy of the vision allele, allowing her to see normal colors, all colors. She also possesses a defective copy of the gene, the allele that would cause color blindness. But because she is heterozygous, she has normal vision. The normal allele is dominant to the defective allele. She is then said to be a carrier of color blindness. Okay? And we're now going to... So let's think about a trait that is X-linked. And here we're going to talk about color blindness. Color blindness is the result of a mutation to a gene that resides on the X chromosome. Okay? So women have two copies of that gene, men have one copy of the gene. Here I've illustrated two parents, a woman on the left, a man on the right, also shown their sex chromosomes and the location of the genes, and then in color indicated which allele they have. You'll see that the woman is heterozygous. She has two different alleles for the color vision gene on her X chromosome. She has one normal copy, one defective copy. She is then a carrier of this trait. The defective allele is recessive, so she will have normal vision, but she carries the allele that confers color blindness. The man, on the other hand, is hemizygous. Doesn't matter what allele he has, he's always hemizygous. In this case, he possesses the normal allele and has normal vision. Now I want you to think about how we would create a Punnett square for a cross of a sex-linked gene. Well, in this case, you would think of the woman, let's put her alleles on the rows of our Punnett square, and the man, we will put the possible alleles that he can give to his offspring. And when we're thinking alleles, let's think a bit larger and think about the sex chromosomes he can give to his offspring. Look and see what. So here I've created a Punnett square for you, illustrating what the mother can give to her offspring on the rows and what the father can give to his offspring in the columns. You will see that the man will pass along his normal allele to all of his, which offspring of his? sons or daughters. Because not only does this Punnett square determine genotypes, it also determines gender. So the first column, the father gives his normal vision allele to all of his daughters, and he gives the Y chromosome to all of his sons. Okay. Now, the mother gives her defective allele to half of her offspring, half of her daughters, half of her sons. And then she gives the normal allele to the other half of her daughters and the other half of her sons. Now I want you to think about the contents of this Punnett square. Who among these offspring will be colorblind? So in squares 3 and 4, obviously those offspring have normal vision. They do not have a single copy of the defective allele. In square 1, we have a daughter, because she has 2 copies of the X chromosome, and one of her copies is defective. She's just like her mother in that regard, and therefore has normal vision, because she has a functional copy of the gene that she got from her father. Okay? So all of the daughters of these parents are going to have normal vision. The son in square 2, unfortunately, has only a defective allele for color vision and therefore will be colorblind. What happens when... Here you can see the results of that cross illustrated with the silhouettes of 4 adorable little children, both daughters having normal vision, one being a carrier because she's heterozygous, and then 2 sons, one of which is colorblind. Okay? These 2 Now, let's dig a bit deeper into a sex-linked question. If I tell you that a man with hemophilia, an X-linked recessive trait, marries a normal woman with no family history of hemophilia, what percentage of their sons will have the disease? What percentage of their daughters will have it, and what percentage of the daughters will be carriers? So when you read a question where you're presented with a scenario like this, your first thought needs to be determining which individuals do you know their genotypes for certain and which do you not know. Okay, what alleles, which genotypes are you uncertain of? In this case, I've told you that the man has hemophilia and it's an X-linked recessive trait. He's hemizygous for hemophilia. He possesses the allele that confers hemophilia. Okay. The mother, she does not have hemophilia, so she's certainly not homozygous recessive. She could be heterozygous, I suppose, but when I tell you that she has no family history of hemophilia, you can safely infer that she is homozygous dominant and does not carry the recessive allele. If you have a question about something like this on an exam, please don't hesitate to ask one of the instructors and we can clarify any doubts. So, what percentage of the sons will have the disease? Well, let's look at the Punnett square that I've created at the bottom of the slide here. Okay? So, where do the sons get their X chromosome? In the scenario I presented to you, the only X chromosome bearing the recessive hemophiliac allele is in the father. Do any of the sons receive an X chromosome from their father? No, because they're only sons because they received his Y chromosome. Okay? What percentage of the daughters will have it? If you look at this, the father is giving all of his offspring that are female the recessive hemophiliac X allele. Okay? But the mother is giving all of the offspring a dominant non-hemophiliac allele on her X chromosome that she gives them. So in squares 1 and 3, you can see that all of the daughters receive a normal functional X from their mother and a recessive hemophiliac X from their father. They're therefore heterozygous. They carry the recessive hemophiliac allele, but none of them have the disease. And so we've just answered the third question here: what percentage of the daughters will be carriers? That is, of course, 100%, as you can see from this Punnett square. In our last sex-linked example, I want to point out something that we haven't yet discussed. And for this, we're going to use a cat example. Big surprise. The genes that determine the coat color of cats, the specific gene that determines whether they are black or orange or some mix of those colors, is found on the X chromosome. So you can see that coat color genotypes, females can be homozygous for B, big B, black, heterozygous or homozygous recessive, homozygous for orange. Males can be hemizygous for black or hemizygous for orange. What's interesting, and we haven't touched upon yet, is the idea of X inactivation. And it's this process that leads to what you see on the left, this confusing half orange, half black cat, which most of you have likely seen before and perhaps not thought critically about until I just told you that there are black alleles and orange alleles for cat color. If that's the case, if it's so simple, how in the heck do you get cats that are partly orange and partly black? Offspring. So, what we're going to talk about now is the concept of X inactivation. So, women and female cats have 2 copies of the X chromosome. Whereas males have only 1, females have 2, but they don't actually need 2 copies of all of those genes. And for that reason, at a relatively early stage in embryonic development, each cell decides to turn off, package, and pack away one of their X chromosomes. They don't need both of them, so one of them is put in permanent storage. You can see this at the top of this illustration, where one of the chromosomes has been condensed and packed away into what is called a Barr body, B-A-R-R. And the most important point to notice here is that each of these cells at this stage of embryonic development has a copy of the X chromosome from this woman's mother and a copy from the woman's father. But the X that is chosen for inactivation is completely random. And in these 3 cells that we've zoomed in on, 2 of them have inactivated the paternal or the blue copy of the X chromosome, and 1 of them has inactivated the maternal or pink copy of the X chromosome. Now, as this embryo continues to grow, and these 3 cells, along with all the others that have inactivated one of their X chromosomes, divide through mitosis, growing the embryo, growing this young girl into an adult woman, each of those cells' descendants has the same X inactivated. So you can see that illustrated below, where each of the 3 has about 10 descendant cells illustrated, and they've maintained the identity of the inactivated X chromosome. What does this mean for cats? What this means for cats is that our heterozygous females that have both the big B, the black color allele, and the small b, orange colored allele, if they are inactivating one of these chromosomes, and their entire body is what we would call a mosaic, a patchwork of clusters of cells that have different X chromosomes inactivated, what we will see then is a cat that has a large chunk of its skin or its surface that is orange next to a big patch that is black. And so those different patches are indicative of which X chromosome has been inactivated. And so you might think now, what does that mean about all cats that have this black and orange coloration? If you look at these cats here, you can see that the mother has both black and orange coloration. We'll not worry for the moment about the white. Okay. So what does that tell you about the genotype of the mother cat? If she has both patches of orange and patches of black, look at the possible genotypes up to the top. Knowing what you do about X inactivation, you know that she must have both the allele for black hair and the allele for orange hair. And what has happened is she has inactivated one of these copies in alternating patches, and she therefore has to be heterozygous. Now, if we look at her kittens, some of her kittens are solid colored. I see a solid whitish cat in there, perhaps, but I definitely see a solid orange cat there, second from the left. So let's ignore those and think about the multicolored kittens. If these kittens have both black and orange, what is their genotype? Well, it's probably just like mom's. But what is their gender? Okay. Now, knowing what you do about X inactivation and the phenotypic expression of both orange and black coloration, you can then infer that those multicolored kittens are not only heterozygous like their mother, but they are also females. Okay. Now, last question. If the mother cat mated with a black male, what's the sex of the orange kittens? Okay, now this is interesting. If the father is black, then you know that his genotype is his X chromosome has the dominant B allele, and then he has a Y chromosome. So, if we have orange kittens, how could they become orange? Well, it's certainly not by the contribution of any X chromosome from the father. The only way you can get an orange kitten is if the kitten receives the orange allele from mother, whom we've already decided is heterozygous, and if that kitten is in fact male, they would have had to have received the Y chromosome from their father. So we know not only the genotype of that orange kitten, but that also tells us the gender of the kitten. Okay, I think we've worked through enough problems now that you should be able to tackle that worksheet that we've posted on Blackboard, and if you can work through those problems and the problems I've presented here, you're going to have little trouble with problems that you encounter on the final exam. Good luck.
Hi, everybody. Dr. Noonan just walked you through cat fur color as a codominant and sex-linked trait. He also introduced you to my two cats, Vinny and Matilda. Matilda is what cat breeders call a tortoiseshell tabby, or a torby. Torbies still have dark black pigment and orange pigment like the tortoiseshells, but show it only in their stripes. You can see it best on the insides of her arm stripes and her belly spots, but the orange pops out throughout her coat. I added arrows by her arms to make it obvious for those non-cat folks out there. Also, Dr. Noonan mentioned that Matilda only has 5 toes. So based on what you know about color and polydactyly in cats, you can try to answer the questions listed here, and I will walk you through them on the next slide. Keep in mind that these are hypothetical, of course, because Vinny and Matilda are both fixed, so can never have kittens. All right, so let's walk through the example. I've animated this slide, so if you click as I'm explaining it, you should be able to see the answers appear as we go through them. For number 1, we know that Matilda has both black and orange pigment. As a female, she has 2 X chromosomes, so her genotype for the pigment locus should be X big B, X little b. Since she only has 5 toes, we know she's homozygous recessive for polydactyly, so we can call it little p little p. This gene is not on the sex chromosomes. For number 2, we'll first need to draw a Punnett square, and if you click, one should show up. Traditionally, we put male genotypes at the top of the square, so on the next click, Vinny's genotype should appear. Since Vinny is black and only has 1 X chromosome, his genotype is X, big B, and Y. We use the big B in this cat coat color example to indicate black pigment. In number 1, you determine Matilda's genotype, so you already know that she should be X big B, X little b, and you can put her genotype along the side of the square. And again, if you click, it should appear. Whenever you go to fill in a Punnett square for a sex-linked trait, don't forget the X's and Y's. When you leave them out, you can make mistakes in estimating the proportions of phenotypes you see in offspring. Now, when you want to answer number 2, it's asking you the proportion you expect to be male and black. You can look at the Punnett square and see that there's a quarter of the offspring that should be X big B, Y. But we also can estimate this a different way. You can estimate that you have a 50% chance of getting a male kitten, and you also have a 50% chance of Matilda giving her big B allele to her kitten, or a different 50% of giving the little b to the kitten. So if you take the 50% chance that you're likely to get a male and the 50% chance that you're likely to get the black allele from Matilda, you end up multiplying those two together, so 0.5 times 0.5, and that tells you a quarter of their offspring are likely to be male and black. For number 3, we can do the same type of math, but since it asks about females, we looked at the left column in our Punnett square. If you look at the Punnett square, you can see that only a quarter of the kittens are expected to be Torby females. So 1/4 of 20 is equal to 5. And the only ones that appear Torby are actually females. For number 4, according to our Punnett square, female kittens can either be black or Torby, so there are no orange females. It actually turns out that it's really uncommon to find a female orange cat, and that's simply because you need to have the 2 little b alleles present to make an orange cat. Now, in the last question, if you wanted to actually look at Vinny and Matilda's chances of having polydactyl kittens, you'd have to know Vinny's genotype. Unfortunately, Vinny will never mate, so we will never know. How many polydactyly kittens they would have really depends on whether or not Vinny is a homozygote or a heterozygote. And if you go back to the slides where Dr. Noonan showed you this, you could actually estimate what the outcomes would be given Matilda's known genotype. And remember, that would actually be an example of a test cross. All right, that's all for this lecture. You can go ahead and start your worksheet now.